Welcome to BioBits and Bytes. Imagine your brain's day-to-day thinking is like drawing a complex network of roads across a vast landscape. If you are struggling with entrenched mental health issues, say chronic depression or addiction, your internal GPS is stuck.
It feels stuck.
You're perpetually forced onto the same deeply rutted negative dirt tracks, making it almost impossible to steer toward a new destination.
Right. And that feeling of being perpetually stuck, that rigidity is more than just a metaphor. It's reflected in rigid pathological neural circuits in the brain. For years, psychiatry has looked for ways to smooth those roads or, fill in the ruts.
Our deep dive today is into psilocybin, a compound long associated with altered states, but which recent science reveals is much more profound. It acts not just as a temporary experience enhancer, but as a powerful, rapid road construction crew.
I like that analogy.
It rips up the old entrenched ruts and lays down new, flexible pavement, facilitating deep and lasting behavioral change.
And our mission today is to connect two distinct, really revolutionary areas of research for you. First, we're going to unpack the clinical evidence from a rigorous trial focusing on alcohol use disorder, or AUD.
Okay.
And second, we're zooming right down to the cellular level using some cutting-edge tools to map exactly how psilocybin physically rewires those neural circuits to create these profound therapeutic effects.
That analogy sets the stage perfectly. Now, if the brain is building new roads, we need proof that this translates into real-world stopping power against entrenched behaviors. So let's start with that clinical data on alcohol use disorder. What was the setup for this trial?
Well, this was an incredibly solid, double-blind, randomized clinical trial. It involved 95 adults aged 25 to 65, all diagnosed with alcohol dependence.
And the design was crucial, right? Because it wasn't just about the drug.
Exactly. All participants received 12 weeks of structured psychotherapy, a combination of motivational enhancement therapy and cognitive behavioral therapy. The real question they were asking was, could psilocybin accelerate or deepen the effectiveness of standard care?
So the intervention itself was delivered against this foundation of traditional therapy, and it was surprisingly brief, just 2-day-long medication sessions given at week 4 and week 8.
Just 2. And participants were randomized into 2 groups. The first received the high-dose psilocybin, ranging from 25 to 40 milligrams, and the control group received an active placebo, diphenhydramine.
Okay, and using an active placebo is a really critical detail. For listeners who might not be familiar, that means they used a drug that actually produces noticeable effects, like drowsiness, to help maintain the blinding.
Right.
You can't just give someone a sugar pill when the other group is going on a full psychedelic journey, or the study is instantly unblinded.
Precisely. They were trying to control for the expectation of feeling something happen. And the primary measure for success was the percentage of heavy drinking days, or PHDD, tracked over a massive 32-week double-blind period after that first dose.
And what did the data show?
The results were, they were genuinely exciting. Over that 8-month period, the psilocybin group reported a PHDD of just 9.7%. Think about that. Less than 1 heavy drinking day for every 10 days observed. That is a massive sustained reduction.
And how did that compare to the active placebo group?
The diphenhydramine group had a PHDD of 23.6%, so that's a mean difference of 13.9 percentage points. That difference is just staggering. It translates directly into months of reduced harm and improved health for patients.
And that reduction is not only clinically meaningful, but it's durable. It lasts for over half a year. Even though the intervention was only 2 sessions delivered months earlier, it forces us to ask, how confident are researchers that the drug was the key and not just an amplifier for the therapy they were already getting?
That's a great question. The statistical analysis showed that the reduction in heavy drinking days was significant above and beyond the improvement seen from the psychotherapy and the placebo alone.
So it had its own distinct effect.
It did. And what's fascinating here is that this isn't entirely new territory. These modern findings are really consistent with meta-analyses of those much older trials from the 1960s using LSD for AUD. This contemporary, rigorous data is just solidifying the evidence.
So that massive behavioral shift, that nearly 13% difference, it must be rooted in some rapid physical change in the brain. We need to know how quickly this rewiring actually happens to create such a long-term change.
That's the perfect bridge. We're moving from the clinic to the laboratory. The convergent downstream mechanism that seems to explain the sustained effects of all classical psychedelics, psilocybin, LSD, ketamine, is neuroplasticity.
Let's pause and simplify that. Neuroplasticity, for the listener, is the brain's ability to change. But what kind of change are we really talking about here?
We're talking about comprehensive structural and functional changes. I mean, the brain is literally rebuilding its physical connections. Researchers now believe that the core of addiction and depression involves a loss of this plasticity, which leads to those entrenched ruts we talked about.
And psilocybin appears to reopen that critical window of plasticity.
Exactly.
So how did researchers actually map this physical rewiring? You can't just peer into a living brain and watch new connections form.
Well, that brings us to some cutting-edge research out of Cornell, which used a pretty sophisticated technique in mouse models. They combined psilocybin with a genetically modified rabies virus, a technique called monosynaptic tracing.
A rabies virus?
Yeah.
That sounds like something out of a sci-fi movie. How is that safe, and how does it help map anything?
It's highly controlled, engineered for tracing only. Think of it like a specialized biological GPS mapping tool.
Okay.
Researchers engineer the virus so it can only jump across a single synapse, a single connection point, and it acts like a tracer, lighting up the neurons it touches. This literally allowed them to visualize and read out the new connections.
The new roads.
The new roads that had been added to the brain's network after a single dose of psilocybin.
That's brilliant. So what specific structural changes did they see? Did the brain just get busier overall?
They observed two very specific, almost opposite changes. First, psilocybin appeared to weaken the cortical feedback loops.
Okay, what are those?
These are the circuits that constantly chatter between different parts of the cortex. They're often associated with repetitive, negative, self-referential thought, the loops that keep you locked in the rut.
So it quiets the inner critic. It throws sand in the gears of that self-critical loop.
Exactly. Second, they saw psilocybin strengthening pathways projecting down to subcortical regions, areas involved in translating sensory perceptions into physical action. It enhances sensory motor responses, making the system more responsive to the external world rather than the internal monologue.
So it's quieting the internal chatter and boosting the connection to the world around you.
Right, but here's the metric that addresses your question about speed. A single dose leads to rapid and enduring structural remodeling.
How rapid are we talking?
We're touching approximately 10% increases in synaptic spine size and density in the pyramidal neurons of the medial frontal cortex. This is a massive physical change, and it occurs within 24 hours.
24 hours.
Within 24 hours, and it persists for at least 1 month.
10% increase in neural connection points in one day. That's phenomenal speed. It suggests the brain isn't just slowly repairing itself, it's aggressively creating a window of therapeutic opportunity. So to pull this together, what's the molecular trigger? What's the key that turns on this rapid road construction?
That key is the 5-HT2A receptor, a specific serotonin receptor. Psilocybin is a partial agonist here. We know this receptor is the central player because if you block it with an antagonist drug called ketanserin, you prevent almost all of the drug's effects, including those rapid synaptic changes.
So the 5-HT2A receptor is the molecular key that unlocks this surge of plasticity.
It is.
That makes the mechanism clear at the cellular level. But the brain is far too complex to assume this drug is a blunt instrument. Does psilocybin just hit all the neurons in the cortex equally, or is it targeted?
Not at all. And that's the nuance that elevates this research, cellular heterogeneity. The research indicates psilocybin is highly cell type specific. They distinguish between two major types of output neurons in the cortex.
Okay.
Pyramidal tract, PT neurons, which are the long-distance communicators sending signals out to the brainstem and subcortical areas, and intratelencephalic, IT neurons, whose axons stay mostly within the cerebrum, connecting cortex to cortex.
So long-distance communicators versus local circuit processors, which ones bore the brunt of the changes?
The IT neurons, the local circuit processors, showed specific alterations, indicating diminished dendritic integration. They were temporarily less receptive to signals, while the PT neurons, the long-distance lines, were less affected.
What does diminished integration actually mean for the function of those cortex-to-cortex circuits? Are they becoming temporarily quieter?
Exactly. Think of dendritic integration as how a neuron sums up all the inputs it receives before deciding to fire. Diminished integration suggests these specific neurons, which are heavily involved in generating our internal narrative, are temporarily going offline, or are much harder to activate.
Which could be the direct neurobiological correlate of that subjective experience of ego dissolution, the loosening of self-referential thought.
Precisely.
That brings us to another level of complexity, the realization that the brain's map isn't static. We need to talk about dynamic functional connectivity, or DFC.
Right. Traditional functional connectivity, or FAC, measures the static average links between regions. But the brain is truly a dynamical, context-dependent system where network configurations constantly shift depending on what you are doing or thinking. DFC captures that fluidity.
And we frequently discuss how psychedelics modulate the default mode network, or DMN, which is associated with autobiographical recall and that self-referential processing.
Exactly. And DFC helps explain how that modulation creates flexibility. One study, using a task switching paradigm, showed a great example of this. When participants encountered increased predictability in a task, researchers saw increased connectivity between regions of the DMN,
Which is usually considered rest mode.
the rest mode network, and networks typically associated with active work, like the control, salience, and dorsal attention networks.
That's fascinating. It demonstrates the DMN isn't just a separate rest center. Its immense processing resources can be flexibly recruited for complex tasks, depending on the need. It really shows the brain's multiplicity.
And we can link this dynamism directly to the subjective experience. In the mouse retrosplenial cortex, or RSC, researchers saw that psilocybin reduced functional correlation among those neurons and, critically, decreased the stability of spatial representations. Trial-to-trial correlations just decreased dramatically.
That is the neurobiological evidence, consistent with human reports of psychedelics disrupting the sense of space and time. Your internal physical map literally destabilizes. The cellular instability maps directly to the disruption you're experiencing.
Absolutely. The whole system becomes destabilized and then rapidly reconfigures itself in a more flexible, less rigid way, supported by that 10% new synaptic growth.
So we have this rapid, targeted, cell-specific rewiring ability, but harnessing it in a clinical setting is the ultimate challenge. How do we move from simply showing what happens in the brain to predicting how to force a therapeutic transition in a human being?
The future lies in computational modeling and building, essentially, digital twins of the brain. Researchers are developing complex models to optimize neuromodulatory therapies. The goal is to move beyond generalized dosing toward personalized, precise interventions.
That sounds incredibly ambitious. What does the development pipeline for this kind of predictive modeling look like?
It's a rigorous three-phase approach. Phase 1 is target identification. You need to identify a specific measurable neural feature that robustly predicts a desired clinical outcome, like reduced heavy drinking. This could be an fMRI connectivity pattern or maybe an algorithmic complexity score like Lempel-Ziv complexity.
What exactly is Lempel-Ziv complexity?
It's essentially a measure of how complex and unpredictable the brain's electrical activity is. A rigid, pathological brain state like severe depression tends to have low complexity because the circuits are repetitive and stuck.
And a healthy, flexible brain has higher complexity, so the clinical target might be to increase that score.
Precisely. So once you have the target, phase 2 is model construction. You build a computational model, often using tools like Wilson-Cowan models, which simulate populations of neurons. These models are informed by the patient's own structural connectivity data from scans like DWI.
And the goal is to tune the model until it accurately generates that target feature you identified.
Exactly. You build a virtual version of the stuck brain. Then phase 3 is optimization.
The engineering phase.
That's it. Once the model accurately replicates the pathological state, you add stimulation or drug parameters, frequency, intensity, dose to the model. The model then predicts the optimal configuration needed to successfully transition that low-complexity pathological state into a high-complexity functional state.
This moves us entirely from observation and generalization to precision medicine, where the dose and the setting could be personalized based on a computational model of the patient's own brain.
And this unified approach means these models can be applied not just to chemical interventions like psilocybin, but also to electrical brain stimulation. We're rapidly moving toward a future where we can intentionally and predictably force the brain to heal itself.
Okay, let's recap the core takeaways for our listener using that road construction metaphor. Psilocybin is a network remodeler. It drives dramatic, rapid synaptic growth. Remember that astonishing 10% increase in spine density in just 24 hours by acting on the 5-HT2A receptor.
And this physical change translates directly into long-lasting clinical benefits, like that major reduction in heavy drinking days we saw in AUD patients. And critically, this process isn't uniform. The key is understanding the context and cell type, like how IT neurons temporarily quiet down.
Which all points to the need for highly targeted future therapies driven by these computational models.
Exactly.
Now, for our provocative final thought for you to chew on, the clinical trials showed powerful and lasting benefits even when patients reported difficult, even unpleasant, subjective experiences during the sessions. If the physical rewiring, that 10% synaptic growth, is happening rapidly and persistently at the cellular level, how much does the intense conscious subjective experience need to align with the underlying biological mechanism for the treatment to work?
That's the question.
Is the trip the context that supports the brain's integration, or is it merely a powerful but ultimately secondary side effect of the chemical catalyst itself? Something to consider as research moves forward.
Always something more to explore in the deep dive.
Thank you for joining again.